Okay, can somebody confirm that you can hear me?
I can hear you.
Thank you. I think it's that one, but let me take a look. That's not it. Let me try again. Ah, there it is. Okay. Let me go ahead and get the worksheet up. Okay, we're doing lab 8 today. Any questions before we begin? All right, if not, let's begin. Like I said, we're beginning lab 8. And that lesson is still converting. Let me close that. And let's go back home. I don't think you're seeing my screen. Are you seeing my screen?
No,
Let me get that. All right, are you seeing my screen now? Anyone?
Yes.
Okay,
Yeah,
great.
I see it.
All right, yeah, the lecture is only 78% done. How do I get rid of that without closing it? All right, let's go to the schedule. Today is February 14th, and we're doing lab 8, identification of the gram-positive. Does that look blurry to you? Or is that just my eyes?
no, it doesn't look blurry at all.
It looks blurry to me, but it might be my eyes today. Don't forget, you should be working on your unknown project. You only have about 2 more weeks to work on your unknown project. Boy, that's scary. Is that where we are? Yeah, we have 1, 2, in 3 more weeks, we'll be getting ready for the final. All right, so that is scary, kind of scary. Oops, didn't mean to bring that up. Let's I guess I should mention your unknown project. Not your unknown project. Your infectious disease project is due this weekend, meaning Saturday, September 18th. Turned it in by 11:59 p.m., Saturday 18th. So get your infectious disease project also done. There is a quiz this week. I should have mentioned that in the lecture. On Wednesday, you should finish quiz 4, which covers lab 7 and chapters 5 and 6. My eyesight is really poor today, so let me blow that up. Yeah, I got that right. Labs 7 and chapters 5 and 6. We just finished lab 6 today. Sorry about that, but for some reason we're a little behind schedule. We should be okay because I've got 3 lessons on chapter 8, although we're supposed to start chapter 14 on February 21st. All right, any question about what we're doing? If not, let's go to the lab. Lab 8, identification of gram-positive organisms. We're going to look at gram-positive cocci and gram-positive bacilli. We'll be specifically looking at Staphylococcus and Streptococcus. Oh, good, the lecture lesson just finished. My computer will now speed up. For the cocci, And for the gram-positive, oh, the enterococcus, that's should be over here. Don't ask me why it's over there. And the Clostridium and Bacillus are actually supposed to be over here, the gram-positive bacilli. This has gotten messed up for some reason. And I think Listeria is in the right place. The Corynebacterium, I'm not sure. I think that's a bacilli. All right, we'll be having some video clips today. You do need to use the gram-positive dichotomous key for the case studies today, and then we'll have case studies. Only use this gram-positive dichotomous key for the case studies in this lab. Do not use it for your unknown project. Do not use it for any other lab. So it's designed specifically for this lab. Upon successful completion of this lab, you should be able to understand the principles and how to interpret the results for the CAMP test, the Bilasculin slant test, the SF broth test, and the Optogen sensitivity test. You will also be using dichotomous key to solve case studies involving gram-positive bacterial infections. When specimens are received by a microbiology lab for routine bacterial cultures, they are inoculated on culture media that will facilitate the growth of common pathogens and placed in physical environments that will support their growth. Microbiologists use colony morphologies and gram stain to provide important information in the beginning classification of the bacterial isolates. However, further biochemical testing is required. In this lab, the CAMP test, the Bilasculin hydrolysis test, the SF broth test, and the Optogen sensitivity test will be discussed. And if you're curious, this still happens today that people run tests to identify the bacteria that's causing an infection in a patient. And when I first was teaching microbiology way back in 2005, I thought that was a bit odd because I would think that they would just sequence the DNA and then determine what species was infecting the patient. But they don't do that. And I'm not sure if it's just much more difficult or it's much more expensive because the identification is done by running tests. Okay. And we'll talk about some of the biochemical tests today. And I know that for a fact because I'm friends with two people who actually are in the industry for doing these tests. Where's my mouse? I lost my mouse. Let me go this way. Oop, that didn't work. Oh, there my mouse came back. The CAMP test is the first test we'll discuss, and CAMP is an acronym for the three microbiologist names who first described the test. So it's Dr. Christie, Atkins, and whoever you say this, Munch-Peterson, CAMP. And what they looked at was the hemolytic effect of some beta hemolytic organisms have when they physically grow near Staphylococcus aureus. You make a single streak of the beta hemolytic Staphylococcus aureus down the middle of a blood agar plate. And perpendicular to that staphylococcal streak, you streak another species to test an unknown organism. And that's shown, for some reason I got an extra page in here. That's shown here, where we have Staphylococcus aureus streaked here, and Streptococcus pyogenes streaked here, struck there, streaked there, whatever that word is. And you notice Streptococcus pyogenes has no enhanced hemolysis near the Staphylococcus aureus. Listeria, on the other hand, streaked here, shows enhanced beta hemolysis in this region near the Staphylococcus aureus. And then Streptococcus agalactiaceae, I can't read that, so I'm having trouble pronouncing it. Streptococcus agalactia, has greatly enhanced hemolysis in this region here on the streak there. Any question about any of that? So for some reason, some bacteria have enhanced hemolysis whenever they're near Staphylococcus aureus. And I'm talking about beta hemolysis. And the hemolysis, for reasons I don't understand, take on the shape of an arrow. Okay, if it's enhanced, greatly enhanced, that's sort of like a slight arrow. All right, any questions about the CAMP test? If not, let's move on to the bile esculin test. Bile esculin, auger slant, is used to differentiate between Enterococcus and Streptococcus. So if you have an infection and it could be Enterococcus or Streptococcus, you then put it on the bile esculin hydrolysis test. And if the organism hydrolyzes esculin, it will make esculatin, which is a dark brown or blackish in color. And this is positive for bile esculin. This is negative. Enterococcus is positive for bile esculin slant, and Streptococcus species are negative for bile esculin. Any question about that? All right. The SF broth test is a shorthand for calling it Streptococcus faecalis, and that's the old name for Enterococcus faecalis. They've now discovered that Streptococcus faecalis is not a streptococcus, and so it's called Enterococcus faecalis, but they still call it the SF broth test. Okay. If you put the organism in the SF broth and the organism grows, if it ferments the sugar glucose, it'll produce acids, and then the purple color of the broth will change to yellow from the acid production. And this is positive for SF broth. This is negative, meaning it keeps its purple color, for SF broth. Streptococcus species are positive for SF broth, and Enterococcus faecalis is negative for SF broth. Did I say that right? I didn't say that right. Let me do that again. Streptococcus species are negative for SF broth, and Enterococcus faecalis is positive for SF broth. Any questions about that? Sorry, I got that backwards. And the Optogen sensitivity test, Optogen is actually an antibiotic, and it is used to differentiate between Streptococcus pneumoniae and other alpha hemolytic streptococci. So what you do is you grow the bacteria on a blood agar plate, and if it is alpha hemolytic and a streptococci, like in the gram stain, you then run the Optogen sensitivity test to determine is it Streptococcus pneumonia from other streptococcal species which are alpha hemolytic. So here we have Streptococcus, which is alpha hemolytic. Let me see if I can blow that up. You can tell it's alpha hemolytic because of the darkening around the bacteria colonies, which are kind of off-white. And here you can sort of see the darkening in this region too, around the bacteria. And then what you do is you put the bacteria on a streak area on the plate, and then you put the paper disc containing the optogen, optogen antibiotic on the area. And then you look to see if the zone of inhibition is 14 millimeters or greater, in which case it is sensitive to the antibiotic optogen. And Streptococcus pneumoniae is sensitive to optogen. And if it is less than 14 millimeters in the zone of inhibition, or there's no zone of inhibition, then you say it is resistant to optogen, in which case it is not Streptococcus pneumoniae, but it is another alpha hemolytic Streptococcus. Any question about any of that? All right. So microbiologists use a dichotomous key to help identify their species, and this uses dichotomous logic. So this will be a repeat of what we talked about. For example, you have the gram stain, that's all bacteria. It's a dichotomous test because you get only gram positive and gram negative cells. And so we have a gram positive, so we scratch this. And then we have gram positive bacillus and gram positive cocci. We have gram positive cocci, so we go this way. Then have gram-positive cocci, which are catalase positive and catalase negative. The catalase test is positive, so we remove the gram catalase negative, and then we go further with the what do we have here? Gram-positive cocci, which are catalase positive. We do the coagulase test. If it's positive, it's Staphylococcus aureus. If it's negative, it's some other Staphylococcus species. It's negative, so it's some other Staphylococcus species. And then we can do the novabiocin test. And I'm not seeing the example here because, oh, there it is. If it's novabiocin negative, it's then Staphylococcus saprophyticus. If it's novabiocin sensitive, then it is Staphylococcus epidermidis. Anyways, that's the dichotomous key. You need to use a dichotomous key for doing the what is that called? The case studies today. You're not going to be physically performing any of these activities. However, you will be responsible for knowing how to perform and interpret them. You can view the following video clips on the SF broth and the bile esculin test, and another clip on the CAMP test. And then review how to interpret the following test by completing this chart. Completing this chart will be part of the worksheet. And let's go ahead and move to the worksheet now. So fill in the test. The first one has been done for you. Gram stain for cocci and bacilli. It's gram positive if it's purple, and gram negative if it is pink. So on the left side column, you're going to put a positive result, and the right side column, a negative result. So tell me what the positive result is for the Schaefer-Fulton stain. And for some of these, you're not going to find it in this lab module. You have to look at a previous lab module. And it may be the earlier lab module, which I believe was 7. Yes. But for the Schaefer-Fulton stain, I think you have to go back further than 7 to find out what the positive results are. Put the answer there, and then the negative result here. For the capsule stain, put the positive result here, and the negative stain there. And you kind of have to describe it, like you can say a halo around the cell or a clear zone around the cell, something like that for the positive. And then do that for the catalase test. I think that was in our last lab. The coagulase test, that was in our last lab. The oxidase test, that was in our last lab. The CAM test, positive result, that's in this lab. The bioesculin test, positive result and negative result, that's in this lab. The SF broth test, this lab. The optogen sensitivity test, this lab. Okay? If you need more information on how to do a dichotomous key, watch the little video on how to develop a dichotomous key. We're not asking you to make a dichotomous key, but this will just help you if you need more help on doing a dichotomous key. And then open the dichotomous key provided in this lab for doing the case studies. We're going to be doing case studies on these bacteria here. And you can get the case studies 1 through 8 and the dichotomous key from the lab 8. So come back to lab 8. Should be in the worksheet. Oh, I'm in the wrong week. There it is. So there's the worksheet for lab 8. And then there's case study 1. Let's just briefly look at it. So we have a patient who's a 13-year-old teenager who attended a week-long summer scout camp. Upon returning home, his mother notices a pustule beginning to develop in his spider bite. The area is warm to the touch and is becoming extremely painful. When her son starts running a fever, she takes him to a walk-in medical clinic for evaluation. The physician orders a gram stain of the exudate from the pus, and a routine culture is done. And here are the findings. So there's the gram stain right there. That's his cells, actually, but the gram stain's right there. And there are 2. And then there's the bacteria on a blood agar plate. So interpret that. And then there's some other tests. They took this bacteria and did the catalase test, and there's the result. And then they did the coagulase test, which that's not stated here. I'm sorry but that's plasma, and you can see after 15 seconds of rocking on the slide, you can see this result and interpret that. You'll have to look in a previous lab for the coagulase test, but you kind of had to do that for the table we just discussed because it's not discussed in this lab. All right, any questions about this case study? And then in the worksheet, put down the species name for what species you think is causing case study 1, and how you do that is you go through the dichotomous key, which is shown I think I've got to back up. Unless it's there. Oh, it's here. Right there. So that's the dichotomous key. So you first start with the gram stain. Is it conchi or bacilli? And then go from there in your dichotomous key. Any questions about how to do that? And let's say it was conchi. I don't recall what it was. Catalase negative. And we'll go. I think this is blood. Oh no, that's this is the hemolytic activity on the plate. I'm not going to tell you what it was, so we'll say it's gamma hemolytic, which is obviously wrong. So then it's Enterococcus or Streptococcus, and then they did the bile-esculin test or the SF broth, which tells you I went the wrong way because the options are not supplied in the case study. You want to look for something that has coagulase, apparently. All right, any questions about how you'd use the dichotomous key? If not, go ahead and work on the lab, and I'm not going to go through the rest of the case studies, but you can find them all here, and there's 8 of them, so it'll take you a little while to get through all of the case studies. Sorry that this is set up as one link for each case study. I thought about putting them all together on one page, but then I realized that the person who made them may give them to me one at a time, like he may change case study 4 and not any of the others, and so I'd have to break them down into the 8 links again. So I decided to just keep it 8 links. Any question about any of that? I think we're done. Let me check to make sure. Yeah, so just write down the species name for all of the case studies. You do generally have to tell me the species name. You cannot tell me the family name, for example. All right, any questions? If not, go ahead and work on the lab. I'll stop sharing my screen.